Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For the section to discuss CRT class 12, the chapter Biotechnology and its applications. Part 2. They are any alternative, the path of our understanding, the genetics can be the show that the farmers may be the obtain maximum yield from the field. So the number of scientists and farmers also they obtain from as a different yield from as in a agriculture. It is their way of to minimize use of the fertilizers and chemicals, their harmful effect on their environment or reduce. So when they get using, what happened to automatically the pesticides will be there decrease as well as any their environmental conditions to get good. Okay, now use the genetically modified crops is possible the solution. So simply the farmers and scientists also they get as an one idea. So when we are using a genetically modified crops, what happened? It is the best solution for as to development of the any crops. Next, plants, bacteria, fungi, and animals. Those genes have been there altered by the manipulation is called as in genetically modified organisms. So simply here, plants, bacteria, fungi, animals. These all genes are altered by manipulation is called as genetically modified organisms. So the genes will be get modified and to develop as new crops. Okay. Next, the genetically modified organism plants has been useful for many ways. Genetically modified as what? They given as a name that genetically modified organisms, their number of uses are there. Let's see one by one. The first one. Here, first one, it made crops more tolerant and abiotic stress, cold, drought, salt, and heat conditions. So when you are using as in this genetically modified crops, they're made of what? More tolerant is present and biotic stress, and it controls as abiotic stress and cold, drought conditions, salt, and heat conditions. Also, the plants they can easily survive. Next, reduce the reliance on any chemical the pesticides, pest resistant crops. So here they improve as in the immunity power means here pesticides will be there decrease automatically, reduce any reliance of any chemical pesticides. Third one, help to reduce as any post harvesting loss. So they are complete to get help as what post harvesting loss means they automatically they postpone their life cycle for 1 week to next week, like. And next, increase the efficient mineral uses plants. This prevent early their extinction of fertility of the soil. So mostly when we are using as this enter the plant, what happened? The plant after the given crop, they are completely destroyed and they are completely they dissolve in the soil to like look like what fertilizer. Next, enhanced nutritional value of food. It contains more nutrition values also present. Like when you observe as a golden rice, it is a vitamin A is rich, that enriched rice. So vitamin A also they having as in a that golden rice. Okay, this is next. In additional to the use genetically modified organisms has been the used as a create in a the tailor made as in a plant supply the alternative resources to industries to form as in that strategies, fuels, and pharmaceuticals. So in this genetically modified organisms, we are applied to as in the different methods like as in strategies and fuels and then pharmaceutics also. Okay, next. Here some of the applications of biotechnology. Here some applications of biotechnology in agriculture that you will study the detail or production of pest resistant plants, which could be the decreased amount of the pesticides used in bt cotton is produced by as a bacterium is called bacillus thuringiensis. So most of the when you studies detail in that in a pest resistant plants, in some diseases will be controlled, we get used as in a pesticides. For example, they given bt cotton, bt means bacillus thuringiensis, simply form in a bt means bacillus thuringiensis. Here bt toxin genes has been there cloned from as a bacteria and they expressed the plant to provide resistant insects without a need the insecticides. So here when we are using as a bt cotton to automatically without using as any their pesticides, they're completely they're controlled to as any crop for about as any the insecticides. In the effect of create in a biopesticides, example when you are using as a bt cotton, bt corn, rice, tomato, potato, and then soya beans, etc. So here some genes are to get modified to get applied to as an entire the crop without using pesticides. The total crop will be there cured. Okay, that is they give an example like bt cotton, bt corn, rice, tomato, potato, and then soybeans, etc. Okay, now let's see cotton. In some strains of Bacillus thuringiensis, the produced proteins that kill certain insects such as in leptopterans, like as in tobacco, birdworms, armyworms, and coleopterans, beetles, and dipterans, foils, and mosquitoes. So these are the different types of insects, like as in lepidopterans, coleopterans, and dipterans. Next, here the Bacillus thuringiensis from as in a protein crystals during as in a particular phase of their growth. So Bacillus thuringiensis, it is get produced as in a crystal proteins in one particular phase of in a growth. These crystals contain toxic their insecticial proteins. So that BT get produced as some crystal proteins. They have as contain toxin insecticial proteins. Why does not toxins does not kill the bacillus? Basically, why they not kill? Because see, actually the BT toxin proteins there exist and inactive protoxins, but once they are insect ingest that inactive toxins, they are converted as an active form to due to as an alkaline pH to gut, which as in soluble crystals. So when the basically the protoxins they having as an inactive condition only, but if in a case once their insect to get once to get insect to get uptake food, it is to get uptake food. Here what happened inside of that mouth to get releasing a saliva, then what happened, pH level will be there increased. Once automatically what happened here, the pH level will be they get increased, then what happened, they come their inactive protoxins to get activated, they get harmful. Okay, next, the activated toxins bind with as in surface of the midgut epithelial cells and create as in pores, causes swelling and lysis, and eventually there causes death of the insect. So then what happened, once the mosquito to get uptake as in a food, what happened in that in midgut epithelial cells here to get create as in a pores and causes the swelling, means pores means make as a small pores holes and swelling and lysis after the blast that entered the midgut epithelial cells. After what happened, the insect will be to get die. Okay now, and here here's some the specific bt toxin genes we are isolated from Bacillus thuringiensis and incorporated into as in a several crop plants such as in a cotton. Here some Bacillus thuringiensis to completely to get their incorporate as in a several crops like as what their cotton crop. And the choice of the genes depends upon the crop and targeted pest as most of bt cotton insect group specific. So mostly the in a targeted pest the most important is what bt toxin, the group of insects they are controlled specific species only. Next, the toxin is coded genes, the cry1 AC is named as cry. Before I say what crystal proteins, right? Cry genes, cry1 AC, cry2 AB like. Let's see here. There are number of them. That example, they are proteins encoded by genes cry1 AC, cry2 AB. These are completely controlled as an A. There, cotton bollworm and armyworm and armyworm. And then here the cry1 AB is controlled as a corn borer, controlled by corn borer. Okay, now these are the incompletely BT cotton. Let's see in the diagram also they given clearly. See cotton bollworm destroyed as in bollworms, fully matured cotton. So before they having like after matured they making as a cotton. Okay, now this is in a completely BT cotton. Pest resistant plants. Several nematodes, parasites, wide variety of plants, animals, including human beings. Here, the nematode, that is Meloidigaina incognitia, that infects the roots of the tobacco plants, causes great the reduction of yield. So here, the plant is that is tobacco. The plant is what? Tobacco. See, in this tobacco plant, that enter the root, there causes as the root causes as in some disease. Some here nematodes, their causes disease, then what happened? The growth will be there reduced. The growth will be there reduced. So how to prevent as in this disease? How to get prevent? Let's see. The novel strategy was adopted to prevent this infection, which was based on their process RNA interference. So here artificially to inject it to as in some genes. Here to get artificially to introduce into as in a genes. There the gene is what nothing but as an RNA interference, simply called as RNAi, silencing mRNA. Okay, see here RNAi takes place. It is all eukaryotic organisms nematode the cellular defense. In that mostly in eukaryotic cells, in this RNAi act as what in a cellular defense. It act as a cellular mechanism. Let's this nematode the involving that silencing the specific mRNA due to as in a complementary double stranded RNA. Basically, single stranded RNA is present. It is participate as in a what in a translation process. But if in case the double stranded RNA is present, what happen? It is complete to get stopped with as in a total the translation process. Here, the specific RNA mRNA complementary DS RNA molecule that binds with as in a prevent translation that completely silencing. So it is get act as a silencing mRNA. It get act as what? Silencing mRNA. The source of this complementary RNA could be form of as infection by virus having RNA genomes in mobile genetic elements transposons that replicates via as an RNA intermediate. So here we are artificially injected to as an genetically modified genes, that is RNAi, that is RNA I once we are introduced, what happened? It get act as in that silencing mRNA, and to get bind with as in a single strand RNA, it's make as what? It finally make as in a double stranded RNA. It make as in a double stranded RNA. Now using agrobacterium vectors, here one vector we are using, that is agrobacterium tuberiferacense, right? Here agrobacterium vector we are using, that nematode specific genes they are introducing into as in a host plant. So here one vector we are using, that vector through we are injected to as in this RNAi. Now this the introduction of the DNA such as to produce both sense and antisense RNA the host cell. So now once we are introducing there that enter the cell to get producing as what? It is producing a sense as well as in a antisense. Produce sense as well as antisense. The RNA cell next this two RNA to bind complementary of each to form as an A, their strand is what? Double stranded RNA. It make as what? Finally, it make as a double stranded RNA. It initiated RNAi thus the silence specific mRNA to nematode. So it get act as what? Silencing mRNA, right? So when the silencing mRNA to get forms as a double stranded RNA, what happened? The translation does not takes place. Now the consequence of was the parasite could not survive in the completely transgenic host expressing the specific their interfering RNA. So it is not further develops. Then what happened? The transgenic plant therefore got as in itself they are protected from as in a parasite. So then finally what happened? It is protected from as in a their parasites. They given here, see here. Mostly this is completely the nematode positive here. These are the nematode. So when we are introducing into as an A, the RNAi, what happened finally here? That purifies entire the disease. Okay, now this is in a completely pest resistant plants in a tobacco plants. Okay, remaining to discuss in next class. Thank you for watching. Please share, like and subscribe. Thank you.